The overall goal of this procedure is to quantify cell senescence in a rapid and sensitive way. Here, we demonstrate the utility of this method in screening potential chemotherapeutical drugs. First, to induce cellular senescence, the chemotherapeutical drug doxorubicin is added to cancer cells. Next, baphyllomycin A1 is applied to neutralize the acidic pH of cellular lysosomes. Then, cell-permeant beta-galactosidase substrate C12FDG is added. Upon cellular uptake, the C12FDG is hydrolyzed and fluoresces. Since beta-galactosidase is enriched in lysosomes of senescent cells, they become more fluorescent than non-senescent cells. To quantify the difference in fluorescence, the cells are run on a flow cytometer. Analysis of the resulting data allows for quantification of the percentage of senescent cancer cells under different treatment conditions.
The main advantage of this technique over existing methods, like the β-galactosidase assay, is that it facilitates rapid and sensitive quantification of senescent cells. In fact, we first had the idea for these methods when we experienced difficulty with quantifying senescent cells using the commonly used β-galactosidase assay. Although we use this technique to detect senescence on cancer cells, it can also be used on normal cells.
This method can help answer quick questions in the cancer field, such as whether a chemotherapeutical drug induces cancer cells to senesce, which might promote malignancy. Thus, this technique could ultimately help identify new therapeutical treatments for cancer.
Working in a chemical fume hood, dissolve the C12FDG powder in DMSO.